Welcome, curious minds, to another deep dive. Today, we're tackling a topic that literally revolutionized modern medicine, but now, well, it faces one of its greatest challenges: antimicrobial drugs. You probably heard terms like antibiotics or maybe superbugs thrown around, but what do they really mean for us, for the future of healthcare? Our mission today is to cut through the noise using the latest edition of Microbiology, an introduction as our guide. We want to give you a clear, concise understanding of how these powerful drugs actually work, why some are losing their power, and what the future might hold. We're going to unpack the major concepts, the incredible microbial processes involved, the structures these drugs target, even some fascinating lab techniques that guide how they're used. And we'll connect it all to real-world clinical and environmental relevance so you walk away truly well informed.
Indeed, it's a deep dive into the very fabric of how we combat infectious diseases. We'll start at the beginning, understanding those historical aha moments, and then move through the intricate ways these drugs interact with microbes. And we'll try not to overwhelm you with jargon. We'll even introduce a compelling clinical case right at the start, actually. A patient with a Pseudomonas aeruginosa infection after a corneal transplant. We can use that to sort of see these principles in action throughout our discussion. So, yeah, let's unpack this.
Okay, you mentioned historical aha moments. I'm fascinated by this idea of a magic bullet, something that could destroy, disease-causing organisms without harming us. Was that just wishful thinking at first?
Not at all, actually. The concept of a magic bullet was coined by Paul Ehrlich way back in the early 20th century. He envisioned a chemical compound that would selectively find and destroy pathogens, but leave the host cells completely unharmed. And that idea really laid the foundation for chemotherapy and this crucial principle of selective toxicity.
Right. And then came that legendary accidental discovery that changed everything. Alexander Fleming in 1928 noticing mold inhibiting Staphylococcus aureus growth on a petri dish. It sounds almost too simple for something so monumental,
It does, but it was a really keen observation. That mold was Penicillium notatum. And the active compound, penicillin, well, it was isolated years later. This phenomenon where one microorganism inhibits another is called antibiosis, which, as you could probably hear, gave us the term antibiotic.
Hmm.
And what's truly fascinating is that much of our antibiotic arsenal today, over half of it, still comes from natural sources, primarily Streptomyces, these filamentous bacteria you find in soil and certain molds too.
Okay, so on one hand, we had these incredible discoveries from nature, like penicillin, but then we also had wholly synthetic breakthroughs, right? Like the sulfur drugs.
Precisely, yeah. Compounds like Prontosil Red, the first sulfa drug, they emerged from systematic chemical surveys, just testing lots of compounds. And this proved we could actually create effective antimicrobials entirely in the lab, not just find them in nature. It really broadened our horizons quite a bit.
So when we talk about selective toxicity, the core idea is hitting the bad guys without, collateral damage to our own cells. How do these drugs actually manage that?
Well, it's relatively easier with bacteria because prokaryotic cells, like bacteria, differ significantly from our human eukaryotic cells. Think about their cell walls, for instance, or the unique structure of their ribosomes, or specific metabolic pathways they rely on that we just don't use. These differences provide lots of unique bacterial-specific targets for drugs.
But the problem gets much harder when the pathogen is also a eukaryote, like a fungus or a protozoan or even a helminth or a virus. It feels like you're trying to hit a target that looks suspiciously like your own team.
That's a perfect analogy, actually. Yeah, these organisms resemble human cells much more closely, so finding a truly selective target is a significant challenge. Drugs often end up damaging the host too, which leads to more side effects. Viruses are even trickier. They essentially hijack our cellular machinery to replicate, so targeting them often means interfering with our own processes. Really difficult.
We also have to consider the spectrum of activity. Some drugs are narrow spectrum, like penicillin G, affecting mostly gram-positive bacteria. Others are broad spectrum, hitting a wider range. Intuitively, broad spectrum sounds better, doesn't it? Like a shotgun versus a rifle.
Well, yeah, it might seem that way. And broad-spectrum drugs can save time if you don't know exactly what you're fighting, but they have a major disadvantage. They can destroy the host's normal beneficial microbiota, our good bacteria. And this can lead to something called a superinfection, where opportunistic pathogens like Candida albicans, that yeast, they flourish because their competitors are suddenly gone. Or resistant strains simply replace the sensitive ones that were killed off.
And this brings us back to our clinical puzzle. Our patient developed that Pseudomonas aeruginosa infection after a corneal transplant, even though they received prophylactic gentamicin. P. aeruginosa is notoriously resistant. Why would a drug meant to prevent infection actually allow this to happen? We'll definitely circle back to this as we explore the mechanisms. So let's dive into how these drugs actually do their work. You mentioned bacteriostatic and bactericidal. Do they always kill microbes or just stop them from growing?
They do both, yeah. Drugs are either bactericidal, meaning they directly kill the microbes,
Uh-huh.
or they're bacteriostatic, meaning they prevent microbial growth. They just sort of pause things, allowing the host's own immune system to take over and clear the infection.
Okay. So what's one of the clearest examples of how these drugs exploit differences between our cells and bacteria? Where's the first place they often attack?
Often, it's that unique bacterial cell wall, a structure we simply don't possess. It's a great target. Drugs like penicillin and cephalosporins, they work by preventing the synthesis of peptidoglycan. That's the essential sort of mesh network that gives bacterial cell walls their strength. Without it, the cell wall weakens and the cell basically bursts. It undergoes lysis. Since our cells lack peptidoglycan entirely, these drugs have very low toxicity for us, very selective.
What about those particularly tricky mycobacteria, though, like the ones that cause tuberculosis? They have a very different kind of cell wall, don't they?
They do. Very waxy. Mycobacterium species have these things called mycolic acids in their cell walls. So drugs like isoniazid or INH, they inhibit the synthesis of mycolic acid. And another drug, ethambutol, inhibits its incorporation into the wall. This makes their cell walls weaker. These are absolutely crucial drugs for treating tuberculosis. It's a great example of really targeted therapy for a specific bacterial feature.
Okay, next up, inhibiting protein synthesis. Now, this sounds challenging because both our cells and bacteria make proteins all the time. How do these drugs manage selective toxicity here?
Yeah, the key is the difference in the machinery, specifically the ribosomes. Prokaryotes, bacteria, have what we call 70S ribosomes. Human eukaryotic cells have 80S ribosomes. They're structurally different, so drugs like chloramphenicol, erythromycin, tetracyclines, they selectively target those bacterial 70S ribosomes. They interfere with the protein-building machinery in different ways. It's kind of like targeting a specific bolt on their assembly line that our assembly line just doesn't have. Ensures our factory keeps running while theirs grinds to a halt.
But are there ever downsides for us, even with that difference? Any spillover effects?
Unfortunately, yes. It's not perfectly clean. Our mitochondria, the powerhouses in our cells, they actually contain 70S ribosomes, very similar to bacteria. It's part of the evidence for the endosymbiotic theory, actually. So these antibiotics can sometimes have adverse effects on us because they affect our mitochondria. Chloramphenicol, for instance, can suppress bone marrow activity, potentially leading to aplastic anemia. And aminoglycosides can cause permanent hearing damage and kidney damage. That's why their use has to be carefully monitored.
Right. Okay, what happens if a drug messes with the outer layer of a microbe, its plasma membrane?
Well, polypeptide antibiotics like polymyxin B can disrupt bacterial plasma membranes. They essentially punch holes in them, causing essential metabolites, things the cell needs to just leak out, kills the cell. For fungi, it's a bit different. Antifungal drugs like polyenes and azoles, they work by combining with or inhibiting the synthesis of ergosterol. That's the main sterol in fungal membranes. Human cells use cholesterol, not ergosterol. So again, that difference provides that crucial selective toxicity. Hit their sterol, leave ours alone.
Interfering with DNA or RNA synthesis. That sounds pretty fundamental to any living thing. How can that be selectively toxic?
It is fundamental, you're right. The selectivity comes from targeting bacterial or viral specific enzymes involved in that process. Rifampin, for example, specifically inhibits bacterial mRNA synthesis. Doesn't affect ours nearly as much. And it's crucial for treating tuberculosis, partly because it penetrates tissues well. Quinolones and fluoroquinolones, like ciprofloxacin, they inhibit bacterial DNA gyrase. That's an enzyme bacteria need for DNA replication, but we don't have the same enzyme. So again, selective.
And what about viruses? We know they're difficult because they live inside our cells. How do antivirals work around that?
That's right. It's a major challenge. Many antiviral drugs are what we call analogs of nucleic acids, meaning they look like the building blocks of DNA or RNA, but they're fakes. When the virus tries to replicate using these fake blocks, its synthesis gets jammed up, blocked. Others target enzymes that are unique to the virus, like reverse transcriptase in HIV or proteases needed for viral assembly. And still others work by preventing the virus from even entering our cells or uncoating once it's inside. Interferons, too, which are signaling molecules our own body makes, can be used as drugs to help inhibit viral spread.
Okay, last major mechanism, blocking a cell's food production or essential chemical reactions, inhibiting the synthesis of essential metabolites.
Exactly, like cutting off a supply line. Sulfonamides, The sulfa drugs are the classic example here. They are antimetabolites.
Hmm.
They competitively inhibit an enzyme in the folic acid synthesis pathway. Many microorganisms need to make their own folic acid, which is vital for making nucleic acids and amino acids. But here's the clever part. Human cells don't synthesize folic acid. We get it directly from our diet. So sulfa drugs inhibit the bacterial pathway, but don't harm our cells because we don't even have that pathway. Clear selective toxicity.
Wow. It really sounds like our antimicrobial toolbox is incredibly diverse with so many specialized tools. Can you give us a sort of quick overview of the key categories and maybe their general superpowers without, needing a PhD to follow?
Absolutely. Let's think of them as different brigades in our fight against infection. First, the cell wall inhibitors. The penicillin family is a cornerstone, obviously. But we also have semi-synthetic versions like amoxicillin, designed to resist bacterial enzymes or hit more types of bacteria. Then you have things like carbapenems. These are incredibly broad spectrum, often reserved for really tough infections. And drugs like vancomycin are critical last lines of defense, especially for multi-resistant bacteria like MRSA. The core strategy here is always disrupting that bacterial outer layer.
Okay, cell walls. What's next?
Then we have the protein synthesis inhibitors. These target the bacterial 70S ribosomes we talked about. They're effective against a broad range of bacteria. Some, like the aminoglycosides, gentamicin is one, have significant side effects, that mitochondrial issue. But others, like macrolides, think erythromycin or azithromycin, are often used for patients with penicillin allergies or for bacteria that live inside our cells.
Got it.
We also have drugs that cause membrane injury, like polymyxin B, which you usually find in topical ointments because it can be toxic if taken internally. Nucleic acid synthesis inhibitors include rifampin, vital for TB, and the fluoroquinolones like Cipro, which are powerful broad-spectrum agents but can have side effects like tendon issues. And finally, the metabolic inhibitors like the sulfonamides we just discussed, often used in combination with another drug, trimethoprim, for a synergistic 1-2 punch effect, especially for things like urinary tract infections.
And the challenges with fungal, viral, protozoan, and helminthic infections, those are quite distinct, right? Fewer truly selective targets means fewer effective drugs, generally.
Precisely. That limited selective toxicity means fewer options and often more side effects for us, the host. For antifungals, the main strategy is attacking that ergosterol in their cell membrane, or sometimes disrupting their cell wall, which is different from bacterial cell walls. Antivirals tend to be very specific to the virus they target, often by blocking entry, replication steps, or assembly. Antiprotozoan drugs, well, you have derivatives of quinine for malaria or metronidazole for things like giardiasis or amoebic dysentery. And antihelminthic drugs for worms, they work by either paralyzing the worms or stopping them from absorbing nutrients. Different strategies for different parasites.
Okay, with all these different drugs and their specific mechanisms, how do doctors actually know which one to pick when an infection strikes? It must be like finding the exact right key for a very specific and sometimes changing lock.
It absolutely is. And tests are essential, especially when susceptibility isn't predictable or you suspect resistance might be developing. The disc diffusion method, often called the Kirby-Bauer test, is very common. You spread the patient's bacteria on an agar plate, then place little paper discs soaked with different antibiotics on top. If the drug works, you see a clear circle around the disc where the bacteria couldn't grow. That's the zone of inhibition. The size of that zone tells you how sensitive the microbe is to that specific drug.
Simple but effective.
Yeah. And then there are more advanced methods like the E-test. It uses a strip with a gradient of antibiotic concentrations. It lets you estimate the minimal inhibitory concentration, MIC basically, the lowest drug concentration that stops the bacteria from visibly growing. And broth dilution tests can go even further. They can determine not just the MIC, but also the minimal bactericidal concentration, MBC, which is the lowest concentration that actually kills 99.9% of the bacteria.
These tests sound absolutely crucial then for preventing overuse, minimizing toxic reactions, and making sure you're using the right drug at the right dose.
They really are. And hospitals also track local resistance patterns using antibiograms. These compile susceptibility data from patients in that hospital or region. They're vital for spotting emerging resistant strains and guiding treatment choices even before specific test results are back for a patient.
Which brings us back perfectly to our Pseudomonas aeruginosa case. The corneas were stored in gentamicin, but the infection still happened. What did the lab tests show? Well, the lab tests later showed that the specific P. aeruginosa strain was highly resistant to gentamicin. It had a high MIC, meaning it took a lot of gentamicin to even slow it down. But the critical factor was the storage temperature. Corneas must be stored at 4 degrees C, refrigerated, to preserve the tissue. But at that low temperature, gentamicin is far less effective as a sterilizing agent than it is at, say, room temperature. Its killing power, the decimal reduction time, was much longer at 4 degrees C. So this long exposure to a less effective concentration of gentamicin at that low temperature likely didn't kill all the bacteria. Instead, it inadvertently selected for any Pseudomonas that were already resistant, allowing them to survive and eventually cause the infection post-transplant.
Wow, so the storage conditions basically created a breeding ground for resistance in this case, which leads us right into the biggest challenge we face today. We're hearing more and more about these superbugs, MRSA, VRE, others that resist multiple drugs. How do these microbes actually fight back against our best weapons? What are their strategies? It's a constant battle, really. Bacteria employ a few core strategies to survive antibiotics. First, they can perform enzymatic destruction or inactivation. Think of it like they have molecular scissors. Bacteria produce enzymes like beta-lactamases. Penicillinase is a famous one that specifically recognize and cut apart the antibiotic molecule, like the beta-lactamering in penicillins and cephalosporins, renders the drug useless.
Okay, so they just break the weapon. What else?
Second, they can use prevention of penetration. This is especially true for gram-negative bacteria, which have that extra outer membrane, like a fortress wall. They can change the little protein channels, the porins, that drugs normally use to get inside, making them too narrow for the antibiotic. Or they can produce those drug-destroying enzymes in the space between their outer and inner membranes, the periplasmic space, so the drug gets destroyed before it even enters the main cell.
Clever, like defending the castle walls.
Exactly. Third, they can use alteration of the target site. This is where the specific molecule or structure inside the bacteria that the drug normally binds to gets changed, just slightly. It's like the drug has a specific key, but the bacteria changes the lock just enough so the key doesn't fit anymore or fits poorly. MRSA, methicillin-resistant staph or aureus, became resistant to methicillin not by destroying the drug, but by modifying its penicillin-binding protein, the drug's target involved in cell wall building. The modified protein still works for the bacteria, but the drug can't bind effectively.
So the target itself changes shape. And the last one.
And finally, there's rapid efflux, or ejection. This is a big one. Bacteria can develop these protein pumps in their cell membranes that actively recognize and pump the antibiotic right back out of the cell, almost as fast as it comes in. Before the drug can reach a high enough concentration inside to actually do its job, it's like having a tiny sump pump constantly bailing out the drug. And what's really concerning is that some of these efflux pumps can recognize and pump out multiple different types of antibiotics, conferring broad resistance.
That sounds incredibly efficient from the bacteria's perspective. And where do these resistance traits actually come from? Do they just appear out of nowhere when we use antibiotics?
Not exactly out of nowhere. They primarily arise from random mutations in the bacterial DNA, just chance variations. But when antibiotics are present, any bacterium that happens to have a mutation conferring resistance has a huge survival advantage. It survives, multiplies, and passes that resistance gene onto its offspring vertically. And crucially, resistance genes aren't just passed down. They can also spread horizontally between different bacteria, even different species. This happens through processes like conjugation, where bacteria directly transfer genetic material, often on plasmids, or transduction, where viruses accidentally transfer bacterial genes. These mobile genetic elements, like plasmids and transposons, often carry multiple resistance genes, allowing resistance to spread rapidly through a population.
So is human misuse of antibiotics making this whole problem worse, accelerating it?
Oh, absolutely. Human misuse is a major driver of resistance globally. It creates the selective pressure. Think about patients not finishing their full course of antibiotics that kills the easy-to-kill bacteria, but leaves the slightly more resistant ones to multiply. Or using leftover drugs for a later illness. Or a big one, healthcare workers prescribing antibiotics for viral infections, like the common cold, where they have absolutely no effect on the virus, but do affect the bacteria living in the patient. All this unnecessary or incomplete exposure encourages the survival and spread of resistant strains.
And what about animal agriculture? We hear a lot about antibiotics being used routinely on farms.
Yeah, that's a significant piece of the puzzle. Estimates vary, but a huge proportion of antibiotics used worldwide, maybe over half, are given to farm animals. And often it's not just for treating diagnosed illness, but used prophylactically, or even just to promote slightly faster growth. This constant presence of antibiotics in livestock creates a massive reservoir of resistant bacteria in animals. And these resistant bacteria, or just their resistance genes, can then potentially transfer to human pathogens, maybe through contaminated food, water, or direct contact. There's evidence linking fluoroquinolone use in poultry feed to the rise of fluoroquinolone-resistant Campylobacter infections in humans, for example. It's a complex web.
It really sounds like it. Okay, switching gears slightly, are there ways drugs can work together to be more effective? Or can they sometimes interfere with each other? It sounds like a delicate dance.
It definitely is a precise choreography. We talk about synergism and antagonism. Synergism is when the combined effect of two drugs is actually greater than the sum of their individual effects. They boost each other. For instance, sometimes penicillin can damage the bacterial cell wall just enough to make it easier for another drug, like streptomycin, to get inside the cell and hit its target, the ribosome. The combination of sulfamethoxazole and trimethoprim we mentioned earlier is another classic synergistic pair. They block two different steps in the same metabolic pathway, making the blockade much more effective.
Okay, so they can help each other. What about antagonism?
Antagonism is the opposite. It's when using two drugs together results in an effect that's less than the effect of the more potent drug used alone. One drug interferes with the other. A common example involves a bacteriostatic drug, like tetracycline, which stops bacteria from growing, used with a bactericidal drug like penicillin, which requires bacteria to be actively growing and building cell walls to work effectively. If the tetracycline stops the growth, the penicillin can't do its job properly. So you generally avoid using those types together.
Interesting. What about just general safety considerations for us, the patients? Every drug has potential downsides, right? A balance of risks versus benefits.
Exactly. Every single drug has what we call a therapeutic index, which is essentially that balance. How effective is it versus how toxic is it? Side effects are always a possibility, and they can range from mild things like nausea or diarrhea to much more serious issues like potential liver or kidney damage or even permanent hearing impairment with some classes like the aminoglycosides. Some drugs can also interact negatively with other medications a person might be taking. Rifampin, for instance, can speed up the metabolism of other drugs, including making contraceptive pills less effective. That's a really important interaction to be aware of. And of course, there's hypersensitivity reactions, allergies. Penicillin allergies are probably the most well-known, but people can have allergic reactions to other antibiotics too, ranging from a mild rash to severe life-threatening anaphylaxis, and always a concern.
Right. So with resistance growing relentlessly, what's next? Are we genuinely heading back towards a time when a simple cut or a common infection could become lethal again, this so-called post-antibiotic era?
There is genuine serious concern among infectious disease experts and public health officials about a potential post-antibiotic era. Developing truly new classes of antimicrobial agents has proven incredibly challenging. The pipeline has been running dry for decades, really.
So if new classes are hard to find, are researchers looking at new targets for existing types of drugs or maybe completely different approaches?
Precisely. That's where a lot of the innovative research is focused now. Instead of just trying to kill the microbe outright, which selects strongly for resistance, researchers are exploring targeting virulence factors. These are the specific weapons pathogens use to cause disease-think toxins or molecules they use to attach to our cells. The idea is to disarm the pathogen without necessarily killing it, which might exert less selective pressure for resistance. Or finding ways to sequester essential nutrients, like iron, that pathogens desperately need to grow, essentially starving them within the host. There's also a critical need for drugs that can combat dormant or persister cells. These are bacterial cells that sort of go to sleep and are tolerant to most current antibiotics, often causing relapsing infections. We need ways to wake them up and kill them, or kill them while they sleep.
And what about looking closer to home? Could our own bodies, our own microbiomes, the trillions of microbes living in and on us hold some answers?
That's a hugely exciting and promising frontier right now. Scientists are actively mining the human microbiome, looking for molecules produced by our normal, beneficial resident bacteria that might act as antibiotics. For instance, researchers found that Lactobacillus gasseri, a common bacterium in the vagina, produces a compound called lactocillin, which inhibits other gram-positive bacteria. And Staphylococcus lugdunensis, found in the nose, produces something called lugdunin, which has shown activity against MRSA in lab studies and even in human trials. The idea that the solution might literally be right under our noses or inside us is pretty compelling.
That is fascinating.
And beyond our own bodies, researchers are looking at antimicrobial peptides from other organisms, things like magans from frog skin or squalamine from sharks. These molecules have been used by these animals for millennia, presumably without generating widespread resistance in their environments. Bacteriocins, too. These are antimicrobial peptides produced by bacteria themselves, often to kill closely related competitors. They're also being investigated as potential therapeutics.
And what about that old idea, the one that was popular in Eastern Europe for a long time, it sort of fell out of favor in the West? Is that getting a second look?
You're probably thinking of phage therapy. Yes, absolutely. Bacteriophages, or phages for short, are viruses that specifically infect and kill bacteria. They are natural predators of bacteria. They were used therapeutically, especially in places like Georgia and Poland, even before antibiotics became widespread, and interest waned in the West with the antibiotic boom. But now, with resistance soaring, phage therapy is experiencing a major resurgence in research and even clinical trials globally. Phages are incredibly specific usually. One type of phage only infects one type or strain of bacteria. This means they can potentially target a specific infection without harming our beneficial microbiota, which is a huge advantage over broad-spectrum antibiotics. There are challenges, like finding the right phage for each infection and dealing with potential immune responses, but it holds significant promise.
It sounds like there are definitely innovative avenues being explored. So finally, let's resolve our Pseudomonas aeruginosa clinical case. The patient got the infection despite prophylactic gentamicin, which tests showed the bug was resistant to. What was the successful treatment?
Right. Given the resistance to gentamicin and the known difficulty of treating P. aeruginosa, the best treatment choice for that patient turned out to be doripenem. Doripenem is a type of carbapenem antibiotic, which we mentioned earlier. Carbapenems have an extremely broad spectrum of activity and are generally very effective against P. aeruginosa, even strains resistant to other drugs. The patient received doripenem and thankfully recovered fully. It really highlights the critical importance of susceptibility testing, understanding these specific drug activities and resistance patterns, and having these newer, more powerful options available in our therapeutic arsenal when older drugs fail.
What an incredible journey through this world of antimicrobial drugs. I mean, from Paul Ehrlich's initial dream of a magic bullet and Alexander Fleming's, serendipitous discovery of penicillin, all the way to the very real and scary threat of superbugs today, and these innovative future approaches like phage therapy. It's clear that this field is constantly evolving. It forces us to be just as adaptable as the microbes themselves, doesn't it?
Indeed, it really does. The mechanisms are complex, sure, but understanding them empowers us. Every single way these drugs work, inhibiting cell walls, jamming up protein synthesis, messing with DNA, blocking metabolism, each has a fascinating story. And conversely, every mechanism bacteria use to resist them, an enzyme that chews up penicillin, a pump that spits a drug back out, is a stark reminder of evolution's relentless power. It's not just about human misuse, though that's a huge factor. It's about life fundamentally finding a way always to survive under pressure. Our challenge isn't just finding new drugs, it's trying to outsmart this incredibly fundamental biological imperative.
That's a powerful way to put it. As you, our listeners, go about your day, maybe take a moment to consider how vital these unseen microscopic battles really are to our everyday health, and how even seemingly small actions like finishing your full course of antibiotics if prescribed, or maybe asking your doctor if an antibiotic is truly necessary, can actually have a huge impact on this bigger picture of public health and preserving these precious medicines for the future. What surprising facts stood out to you today? What connections did you make to this ongoing biological arms race that medicine is constantly fighting?
Keep those questions coming, keep thinking critically, and remember, there's always more to learn about the unseen world all around us and inside us.
Thank you so much for joining us on this deep dive. Until next time, stay curious and keep exploring the fascinating world around us.